Welcome to our first supplementary lecture in biochemistry. For lecture number 2, we discussed some elements of organic chemistry, namely molecular structure and stereochemistry, but I thought that I would take the opportunity now to tell you in video about some organic chemistry that you might have forgotten from last year. So these slides that you see here coming across the bottom are available on the Moodle site. You can go through them, and let's start our exploration of organic chemistry, functional groups, and reactions that you will recall from last year's organic chemistry. Now, here we go. Part 1, functional groups. So alkanes are a class of molecules that I would argue have no functional groups. If you look at your alkane here, you'll see that basically there is nothing here that is fun. It is all carbons and hydrogens. Everything's very nonpolar. There's really not a lot of reactivity here. These make great solvents for organic chemical reactions because pretty much they're mostly unreactive. So alkanes are your sort of basic molecule, which we can decorate with functional groups. What kind of functional groups could we see added to alkanes to change their functionality and their chemistry? Well, we could oxidize it, removing 2 of the hydrogens and a pair of electrons, and have a double bond. Double bonds have a different molecular orbital structure. They're very reactive compared to alkanes especially, and they're planar. They don't rotate around the double bond. A lot of interesting properties to alkenes. And we can see the kind of chemistry that can happen around this double bond a little later on in this lecture. Alcohols. Well, you know, when we say the fun and functional group, this must be the molecule that defines all that. But alcohols are an alkane with a hydroxyl group attached to one of the carbons. Now, this is a highly polar bond, and that's going to give it a lot of interesting properties. It's going to help this molecule interact with water, and there's a lot of chemistry that can happen around that oxygen. It can be oxidized very easily. Here's the different kinds of alcohols. The first one was a primary alcohol. This is a secondary alcohol. This is a tertiary alcohol. And if you want to sort of, I think you can see what primary, tertiary, and secondary mean. Primary is when it's off the end of a molecule. Secondary is when it's in the middle of a molecule with 2 carbons on either side of the alcohol group. And tertiary is when there's 3 carbons on either side of the alcohol group. And so basically you can see it basically just comes down to the number of hydrogens attached to the carbon of the alcohol group. So primary, definitely at the end of a molecule. Secondary, in the middle of a molecule. Tertiary, also in the middle of a molecule, but everything around it is carbons. Amines are a very important functional group. They're a half of an amino acid, aren't they? They're a nitrogen instead of an oxygen. Again, a very polar bond. Different number of hydrogens because nitrogen has a different valency. This is a primary amine here, and this is a secondary amine. A secondary amine is the nitrogen has 2 carbons attached to it. Notice how that's different than an alcohol. Now remember, this is a hydroxyl. An alcohol is actually the carbon attached to the hydroxyl. It's that CO group. So when we say a primary alcohol, we mean, look, 1 carbon attached to the alcohol group. What's the alcohol group? It's the C and the O. So an amine group is the nitrogen here. So when we say primary, we say 1 carbon attached to nitrogen. When we say secondary, we say 2 carbons attached to nitrogen. Tertiary would be 3 carbons attached to nitrogen. With the alcohol, it's 1 carbon attached to the alcohol group, 2 carbons attached to the alcohol group, or 3 carbons attached to the alcohol group. Thiols are just alcohols with a sulfur instead of an oxygen. And of course, they're famous for stinking, and that's a burden they must bear, but they also have interesting chemistry. Sulfur is in the same period as oxygen, so it's got a lot of the same chemical properties, but it is a much bigger atom, and this changes the nature of molecular orbital overlap, and basically sulfur is effectively more electronegative to the carbon next to it than oxygen because it is electronegative like oxygen, but it can't back donate electrons from its orbitals as well as oxygen can. So it's net more electron withdrawing. And that has a lot of properties that can be conferred to biomolecules, and we'll see that in how coenzyme A operates, why we chose to use a sulfur rather than an oxygen. So those are some sort of major functional groups that involve single bonds. Here's some that involve some double bonds to heteroatoms. This is an aldehyde. It's a carbonyl group, but the carbon is at the end of a molecule. You know, it ends with a hydrogen here. So that's an aldehyde. Very reactive, generally, aldehydes. Ketones, slightly less reactive version of a carbonyl group. There is a carbon on both sides. So an aldehyde is at one end of a molecule. A ketone, of course, is a carbonyl group that's in the middle of a molecule. You can easily oxidize an aldehyde to a carboxylic acid. You can't oxidize a ketone to a carboxylic acid. Easy for that proton to be gotten rid of. You can't get rid of a carbon so easily. So we can easily oxidize an aldehyde to a carboxylic acid by just adding water and then oxidizing that. Carboxylic acids, of course, are acidic. That proton can easily be accepted by water or any other base and leave this behind as a negatively charged carboxylate salt. Esters are a condensation of carboxylic acids and alcohols, and basically it is this carbon and two oxygen groups, double bond to oxygen, single bond to oxygen with carbons on either side. We can easily turn carboxylic acids and alcohols into esters. We can easily chop them back to carboxylic acids and alcohols. So it's a great way to create a linkage between an alcohol and a carboxylic acid. And we'll see how it's used to make a link between fatty acids, which have carboxylic acids at the end, and glycerol, which has 3 alcohol groups when we build triglycerides and phospholipids and things like that. You can make esters with sulfur. That's a thioester. It's more reactive than an ester, and we'll see how that's used later on in biochemistry. Amides, a really important functional group. They make up proteins. So if there's a sort of a linear strand of protein made up of all 20 amino acids. I tried to express all 20 amino acids along there. It is a carbonyl group attached to a nitrogen. It has different properties than an oxygen, as we shall see. The nature of this single bond is very different than the nature of this single bond in esters, and that has a lot to do with the structure of proteins and life itself on Earth. So, some more functional groups to talk about. Amines are a condensation of an aldehyde or a ketone and a nitrogen. And this double bond N is easily hydrolyzable back to the aldehyde and the amine it came with. But notice how, look, what I've done here is I have connected a amine with an aldehyde through that double bond there. So amines are great ways to make connections between nitrogen-containing groups, of which proteins are very rich, and aldehydes. And it's a very important part of protein structure and in chemistry, in biochemistry. Ethers, that is a single oxygen between 2 carbons, oxygen that bridges 2 carbons. And this condensation of 2 alcohols is how we might make an ether. You recall that from organic chemistry, probably one of the first reactions that you talked about in organic chemistry. Ethers create this link through oxygen, and there's of course some sort of different flavors of ethers that are important in biochemistry. One is the hemiacetal. That is an ether with an oxygen group, and it's the result of an addition of an alcohol to an aldehyde. There is also acetals, which is when we condense another alcohol with this alcohol group right here to create basically a diether. There's 2 ether connections to a single carbon. How do you spot an acetal? Look for a carbon that has 2 oxygens attached to it. And it might be referred to as a hemiacetal if one of the oxygens isn't an ether, it has a hydrogen. H for hemi, H, hemiacetal. No H there, that's a carbon, regular acetal. And of course, ketones have the same thing that can happen. You can have a hemiketal and you can have a ketal. And that's just basically the idea of a ketone with an alcohol added and then condense that alcohol group here with another alcohol to create the diether. And these hemiacetals and hemiketals to a lesser extent, but hemiacetals are the link that we see in starch. So there's basically actually I believe this is cellulose. So I've presented here the structure of cellulose. And where is the hemiacetals here? How can I find them? Look for a carbon. There's a carbon with 2 oxygens attached to it. That's the hemiacetal link right there. Where's another one? Right here, a carbon with 2 oxygens attached to it. If you look for any of the other carbons, this one here only has 1 oxygen on it. This one here only has 1 oxygen on it. This one, 2 oxygens. Well, now how about some different functional groups? Phosphates. I see no carbons there whatsoever. I thought this was like biochemistry. But of course, as you may know, phosphorus is a very important molecule or atom in biochemistry. That's just a phosphate salt. It's a phosphoric acid. If it was all hydrogens here, that would be phosphoric acid. This would be a phosphate salt. You can imagine 2 sodiums taking up those positions. We can make an ester. Remember, a condensation of an acid and an alcohol is an ester. Well, there's phosphoric acid. Imagine methanol here condensing to create a phosphate ester. So there's a methyl group, there's the phosphate group. This is an ester linkage between a phosphorus and a methyl group. And now, of course, you could do two of those. So now I've got a phosphate diester. And you know what? That is the connection in DNA. Another thing that can happen in organic chemistry is we can have a condensation not of an acid and an alcohol, but of two acids can come together. And so there's one acid, there's the other carboxylic acid, and notice that there's an oxygen bridge between them now. That is a very reactive molecule. We're not going to see too much of that in biochemistry, but what we will see a lot of is phosphoric acid anhydrides. Look at that anhydride linkage between these two phosphoric acids. Look, here's a double anhydride. There's two anhydride linkages between these phosphoric acids. Well, that's the business end of ATP. Very high energy molecule and a building block of DNA. And remember that DNA was all these phosphate diesters? Well, there's DNA right there, and all of these orange atoms here are phosphorus, and there's that chain of phosphate diesters that connects DNA, all the nucleobases together in DNA, or nucleotides, I should say. So those are some important functional groups, their structure, and as we get to know them, we'll get to see their reactivity. So let's just start off with some organic chemical reaction. And most of these reactions have their analogous reactions in biochemistry, usually done a little differently because obviously biochemistry is all done in water. But let's take a look at some of the reactions that we remember from last year in organic chemistry. Remember those double bonds? I said they could be added to. Watch as I add water across this double bond here, and I get an alcohol. And that's a reaction that we will see in biochemistry, the addition of water across a double bond to give an alcohol, and the reverse of it to return us back to a double bond. So I can add things to double bonds. There I'm adding water to a double bond. There's the OH group of water added, there's the other hydrogen of the water added, and of course, if I'm doing that, I'm changing the double bond to a single bond. So you can add things to double bonds. Now, you can do the exact reverse. I can eliminate water and create a double bond. So those are two reactions that we need to remember from organic chemistry. Now, if I can add to a double bond here, I could add to a double bond that's a carbonyl. Carbonyls are much more reactive than alkene double bonds. It generally requires extremely strong acid conditions to do this in a test tube. How can it happen in biology? We can't have strong acid in our blood cells. Or in our epithelial cells, and yet this reaction is happening in every single cell in your body that can do respiration. These two reactions are happening at pH 7. So that's part of the amazing catalytic activity of proteins. But what's much more easily able to be accomplished even in water at neutral pH is the addition of an oxygen atom to a carbonyl. And so here I'm adding an alcohol to an aldehyde, and I'm creating that hemiacetal. And here I'm adding an alcohol to a ketone, and I'm creating that hemiketal. So I can add things to double bonds, add things to carbonyls, add things to alkenes. So keep that in mind. I talked about condensation reactions a lot as we were talking about functional groups. I said that esters were condensation between carboxylic acids and alcohols. Amides were condensation between carboxylic acids and amines, things like that. Well, here's some condensation reactions, and notice what they all share. They all produce water. That's why they're called condensation reactions, because water is produced. And now if I was an organic chemist, I would do this in a solvent like, well, let's go old school and do this in benzene. And then as I boil this, the benzene water azeotrope would form, and it boils actually at a lower temperature than the benzene itself, and I can distill it off, taking the water with it, always pulling the reaction forward because one of the products is being pulled away. That won't work in biochemistry because water is the solvent of biochemistry. It'll be interesting to see how that problem is solved. How can I drive these reactions forward when really, if this is the solvent, it should be really being pushed backwards. And I'll show you how that is done, mostly with the power of ATP. But notice all of these reactions, that OH and that OH, we're going to combine them, and there's one of the oxygens, and we've eliminated water. So condensation reactions are two things coming together and eliminating water as a side product. And that's where we're going to get our amines, our esters, our amides, our thioesters, our phosphate esters. And here is an example of taking a hemiacetal and condensing it with an alcohol to create the acetal. And this would be how I would convert a single sugar into a polymeric sugar, like starch or cellulose. Now, I sort of hinted that ATP was used to power these reactions. I have to get rid of water. What's one way to get rid of water? React it with ATP. Look at this. If I react water with an acid anhydride, I'm going to get the acid back, and the cleaved product. What happened to the water? It's now part of the acid. Now, we're going to use sort of an analogous approach. I like to think of ATP as nature's dehydrating agent. It's being used to drive forward these condensation reactions by, well, taking the role of absorbing the water. It doesn't really absorb the water. That's a completely incorrect analogy, but what it's doing is it's acting like it is. We're actually, instead of using water or allowing water to come off, we're going to react the phosphate with the alcohol before we add the next molecules, let's say the second alcohol. And so instead of it being a condensation reaction that produces water, it would be a substitution reaction that produces phosphate. So we're going to see how that goes in biochemistry. Now, the opposite of condensation is hydrolysis. This is the exact opposite of all the reactions I presented on the other slide, just written backwards, really. Water plus an ester will give you the original alcohol and the original carboxylic acid. This is how things go in the body in the absence of anything else. There's all kinds of enzymes that will cut amides, cut proteins up. They're proteases, and all they do is they take the water that's all around and they just allow this reaction to go the direction it wants to go, because water is the solvent. It's going to want to push things this way. If I want to go in the opposite direction, it's going to take the use of ATP and clever biochemistry to sort of mimic or fake that dehydration process. Substitution reactions. Well, here's an example of activating an alcohol using a phosphate. If I activate that alcohol with this phosphate, then interesting chemistry could happen after that. So this is a very high energy molecule. It releases quite a bit of energy to go to a shorter anhydride, a lot less negative charges around each other. So this is really a one-way reaction. And then one-way reactions are a great way to drive pathways forward. If you've got a biochemical pathway, if you put a couple of one-way reactions in it, the whole pathway is only going one way. If you have a big line of people in a crowd and the people at the front and the people at the end are all going in the same direction, everyone in the middle is going to have to go that way too. So ATP is used to drive pathways, and here's an example of how it could do that by transferring a phosphate to intermediate in the pathway, and that's a one-way reaction. And then we could take that phosphate, and I could do a nucleophilic attack because phosphate's a pretty good leaving group, and now I produced an amine. So that would be making an amine. You could imagine it was a condensation reaction where that was kicked off as water and the nitrogen took its place. But here what I've done is I've phosphorylated, then substituted. And we'll see that this is how we drive those condensation reactions. Phosphorylate, then substitute. And the net product is, a cleaved ATP, as if ATP had just reacted with water. But what we really did was we phosphorylated an alcohol, or at least an oxygen atom in a molecule, and then we replaced that whole group with something else. Isn't that neat? So activation by phosphate is a major sort of idea that we'll see throughout biochemistry, and there's sort of the 3D structure of ATP. You've got the adenosine, you've got the sugar ring, and you have the triphosphate, and we'll be talking a lot about this molecule. You'll get to know it really well. Oxidation is an important reaction in biochemistry. Here's an example of oxidizing an alcohol to an aldehyde, or a single bond to oxygen to a double bond to oxygen. Carbon loves maximizing its bonds to oxygen. I mean, really the ultimate would be to make CO2 out of everything, right? And that's a carbon with 4 bonds to oxygen. And that's why wood burns. That's why everything moves forward to CO2 that's made out of carbon. And that's really the major source of energy for living cells, is taking carbon-containing compounds that we've eaten and convert them ultimately to carbon dioxide and extract all of these electrons, which can be used to drive reduction reactions that want to happen and extract energy from that and build biosynthesis with this. Like, we can make stuff with these electrons because you need electrons to make bonds. If I want to connect two things together, I need a bond. Where am I going to get the electrons from? This reaction right here. And so here's oxidizing. That was oxidizing a primary alcohol to an aldehyde. Here I'm oxidizing a secondary alcohol to a ketone. You can't oxidize tertiary alcohols because you could see I've got to get rid of that hydrogen as part of the process, and if that was a carbon, we couldn't get rid of it. Here I'm adding water to an aldehyde to create this hydrate of an aldehyde, and then what am I doing? I can oxidize that to a carboxylic acid. That's how you oxidize aldehydes to carboxylic acids. Hydrate it, and then oxidize the hydrate product. And again, electrons are kicked out during the oxidation process, and these will, of course, be riding on NADH and moving forward through metabolism to generate energy. All right, so another important thing about carbonyl compounds is that they can rearrange a bit. You just have to move 2 protons. You just have to do a little acid-base chemistry and move 2 protons, and that's easy to do in water. Any aldehyde or ketone will be in equilibrium with a small amount of what we call the enol product. Notice that's an enol. And so the enol product is a very electron-rich double bond. The lone pairs of this oxygen are in resonance with the whole system here, and they're going to really help push electrons out of that carbon. Same is true as if I take the proton off of this hydrogen here to give this enolate, it's exactly the same as if I had taken the proton off of the carbon. So enolates represent a very nucleophilic carbon atom. Enols are also a nucleophilic carbon atom because the electrons are going to get pushed out of the double bond through that carbon. So you'll recall from your aldol additions, your Claisen condensations, those reactions you learned about last year, that's what enols and enolates do. They're going to create a nucleophilic carbon, and this is going to be the basis of how acetyl coenzyme A puts that carbon group into carbon-containing molecules. It's going to use these enol and enolate nucleophilic carbon strategies. So that's going to be a reaction we're going to learn a lot more about. Here we see it in action. There's our enol. Electrons are pushing down from the oxygen lone pair through this double bond carbon into an adjacent aldehyde. And this is the aldol addition reaction. Notice I've made that new carbon-carbon bond. What do you do with new carbon-carbon bonds? You just made a bigger molecule. Maybe we're on our way to making a big, long fatty acid there and then storing it away as energy. You got to put your Christmas somewhere. So that's one way you can store away all that energy from all that food you ate over the holidays. We talked about thioesters. Well, there's an example of acetyl coenzyme A, and it's a thioester. And because I said sulfur is a little more electron withdrawing than the oxygen of an ester, it's going to help stabilize this enolate form. And so the enzymes that are involved in using coenzyme A are going to be able to make these enolate intermediates a little more easily than if it was an ester. And so obviously you can see how that carbon is a lot more activated to be a nucleophile when it's attached to a sulfur thioester compared to a regular ester. So that's one reason why we use thioesters in coenzyme A rather than a regular ester. So here we are adding acetyl coenzyme A to oxaloacetate. This is the first step of the Krebs cycle, and here we have made citric acid basically. And there's the new bond, this dark blue line right here. The rest of the one molecule is on the right. The acetyl groups, the acetyl group's on the right. The rest of the molecule is on the left here. Amides, very special molecule. The nitrogen, just like an oxygen, has a lone pair, but oxygen is more electronegative than nitrogen. So if I was to donate these lone pair into this single bond here to make it a double bond and kick electrons up to the oxygen to create this resonance structure here, this is actually more stable than it looks because oxygen loves electrons. So actually, if these were two oxygens, they're not going to exchange electrons much because, you know, one oxygen is the same as another. But if the oxygen is more electronegative than the nitrogen, it's taking the electrons from the nitrogen, stabilizing this structure, and look at that double bond there. If that single bond is a double bond some of the time, well, it's a double bond some of the time. If it's more than a single bond, it can't rotate. It kind of looks like this really, if you think about it. It's not just a plain single bond. It's a distributed electronic system. Now, to obtain the stability of this resonance structure, you have to have all the atoms that I'm showing you here. They all have to be planar. This oxygen, this carbon, and that carbon, this hydrogen, this nitrogen, and that carbon there. They have to be in the same plane. Otherwise, the electrons can't flow like this. Now, if they weren't planar, if everything was sp3 tetrahedral, that's fine. You just can't do this resonance. But if they were planar, everything sp2 hybridized, then you do get this resonance. Both structures are possible. This is just more stable. So we see that the nitrogens in amides are planar, sp2 hybridized, with a free p orbital to overlap with the pi system of the carbonyl group here. So because they're flat, because they're planar, because they don't rotate easily here, that has a lot of impact on the structure of proteins. And we're going to be talking about that a lot later on in this course. Now I said there was this like electron distributed system here. There is the lowest energy molecular orbital of this system. And you can see, look, it's 3 p orbitals that have all lined up side by side and overlap with each other. And you can only do that if you have 3 p orbitals. And of course, there's a p orbital for each of the atoms of the double bond. How do you get a third p orbital over here? Make that nitrogen planar, sp2 hybridized. sp2, wait a minute, shouldn't there be 3? What happened to that third one? It's here, right here, sticking straight up out of the plane and now overlapping with the pi system. So if we have an sp2 hybridized nitrogen, we can do this and get this nice stable system. And you can see because of that overlap, electron density flows through all three atoms. I can't rotate this bond very easily. Aromatic systems are somewhat similar. You look at benzene here. Every single atom here is sp2 hybridized, which means there's a p orbital coming straight out of the paper at us for each and every one. And we often draw benzene like this. We say it could look like this or it could look like this. But that's a resonance system. It's not one or the other. It's both at the same time. We often draw it like this to express that everything is happening at the same time. Resonance isn't it's sometimes this and it's sometimes this. Resonance is when you say it's both at the same time. And so we have electron distribution. We don't see a pi orbital and a separate p orbital some of the time and this overlap system some of the time. We see this all of the time. And so it's like all of the time we have overlap. If you're going to say it's double bond, single bond some of the time and single bond, double bond some of the time, then you'd have to look like this. It would look like that's a single bond and some of the time it's a double bond. But a double bond is shorter than a single bond. And so you'd have to have the atoms moving. This would have to be two vibrational structures. So the atoms kind of, the atoms on either side of this bond going long, short, etc. But we don't see that. We see this. All the bond lengths in benzene are exactly the same. And guess what? They're at an intermediate length between a single and a double bond. It's not double bonds turning into single bonds and back into double bonds. It's everything exists simultaneously. Cross your eyes, let these two structures blur over each other, draw it like this. That's what resonance is. And it looks like this. All the electrons are overlapping. And that can only happen if everything is coplanar. So if we have resonance, you've got to have p orbitals that can overlap, which means things have to have planar structures. That's very important in biochemistry. Not only is it important in the planar nature of amides, which was a major conceptual hurdle chemists had to get over. One of the reasons that the structure of proteins wasn't figured out until the 50s was because resonance was not understood by biochemists until Linus Pauling taught them all about it in the 1950s. He was telling people about it since the 1930s, but it took a while before his interests fell toward the structure of proteins, and when they did, he quickly realized that amides were planar in proteins and was able to produce the structure of the alpha helix just by knowing the true structure of an amide, when no one else could, despite all the scientific data that was present. Well, here is a tetrahedral nitrogen. Is it that? Is that the structure of an aniline? Or is this the structure of aniline, a planar system with these lone pairs are in a p orbital, and the p orbital is now lined up with all the p orbitals of this benzene ring, and maybe we could get some resonance going. If that resonance is happening, all these atoms must be in the same plane so that we can have a spare p orbital here. The nitrogen must be sp2 hybridized. Well, guess what? Aniline is sp2 hybridized. If I can distribute the electrons, I'll have a more stable system. That's really important in DNA. Take a look at this sort of structure. It's, what would that be? That would be uracil. So here we've got a uracil, a cytidine. That's a cytidine, my mistake. Uracil has two carbonyls here. That's a cytidine, and you've got this nitrogen here. It's planar. Every atom here is planar. The key thing is that that nitrogen is planar. Look at that lone pair. You might think, your Vesper instincts might say make that tetrahedral. But it's planar because of resonance. You can see how you can generate this resonance structure. Here is the molecular orbital of cytidine. Notice how it's all spread out. And because that p orbital here is able to donate into the system, this nitrogen is planar. Because we've got this flat planar face here, I can interact well with the flat planar face of all the other nucleotides, which are also feature planar nitrogens. Because they're flat as a board and their edges line up like the teeth of a key, you have that nice recognition A to T, C to G that you have in DNA. So these planar elements are important in DNA and they're important in amides. So resonance is something that we're going to want to recall. All right, so as you go through this lecture, I want you to try and recall these points. You've got to think about the structures of these functional groups and think about their shapes. Think about the chemistry that you remember from last year. How is an alcohol different than an aldehyde? Try and recall that. So basically, review your chemistry. You don't have to think that you've got to memorize your organic chemistry from last year right now. We'll be going through all of these reactions as we explore the course, but just expect that on a regular basis, you're going to see reactions from organic chemistry, and I will show you how they occur in biochemistry, and we'll explore the chemistry then. By way of my program of introducing you to many more aperitifs in this course, this is Campari. It's one of the first aperitifs. It was an Italian concoction. It's basically a wine infused with all kinds of herbs and spices. And it is one of the first alcohols that featured sort of an aggressive advertising campaign. Gaspare Campari here, who was the son of the founder, hired edgy modern artists like Dudovich here to create these racy posters. And if you look at a Campari ad today in a magazine, in a glossy magazine, there'll still be racy ads. So marketing was really important. And remember, biochemistry is all about coming to understand how molecules affect biology. And if you think back to what I said in the early lectures here, how the early medicinal tonics were the sort of the beginning of these aperitifs and digestives here, Campari would have been one of the early ones. It would have been intended as a sort of to open up the taste buds ahead of a meal or to calm the digestion after a meal. Basically, it's not the kind of drink that you drink on its own. It's a drink you have a sip of before you do something else. It has a purpose. So try to review your organic chemistry and get ready for the organic chemistry that you're going to see in biochemistry.